I think what's been interesting about the development of immunology has been that in many areas in the past, in the early 1900s, people worked alone and in their own field without sort of reference to what we now would call multidisciplinary work. And I think immunology is not in a cocoon on its own. I think you need the interplay between chemistry, biophysics nowadays, biology, microbiology, and so on. So I think that's really important. I think also that scientific dogma can stimulate in the various areas, scientific dogma in chemistry and microbiology and so on, can stimulate scientific advances, but it can also, of course, the dogma can also hold advances back by creating sort of artificial barriers, as it were, that this is how it is and, you better believe it, sort of thing. So I think there's some good lessons in immunological history along those lines. An example perhaps would be Paul Ehrlich. Many think the father of the magic bullet idea, one of the fathers of immunology. When he was developing the idea of antibodies or antitoxins, as he called them, he recognized that antibodies could react with our own proteins, our own human proteins, sort of generating autoantibodies. But actually, he pretty firmly stated that he believed there were physiological mechanisms that prevented autoimmune disease. And some other historians, Arthur Silverstein being one, has suggested that this probably held back the field of autoimmunity research for maybe even decades. So I think that's another example from history where I think strong scientific opinion sometimes can hold back, although of course it can also advance things. What I think has been good in immunology history, there have been some very good, vigorous debates backwards and forwards. And I think immunologists have generally, understood that at any point in time, knowledge is bounded. You can only know what's within your experience. And so things that, when we look back, we think are rather odd that certain people believed, certain mechanisms in immunology. Of course, at the time, that was the only knowledge they had. So I think we also have to recognize that today we think we have the answers, but actually 10 years or 15 years or 20 years from now, other immunologists will look back at us perhaps and say, that was a rather strange conclusion these guys came to. I think there are many examples of that, and of course we can't be critical of that because, as we've just said, the knowledge at the time generates, of course, the dogma of the time. Good example, however, is perhaps in the early part of the 20th century, in the 1900s, when Pasteur was fighting against what you might call the chemistry establishment. The idea of spontaneous generation of living organisms coming from a sort of organic soup. And Pasteur didn't believe that, even though, of course, he was up against the enormous chemistry establishment. But he pursued that, he persisted with his experiments, and during his work on fermentation by yeast, he pretty much firmly established the so-called germ theory, that fermentation didn't arise, because of molecules that happened to be present interacting with each other, but it was actually organisms, microorganisms that actually were responsible for this. And that took the field forward, but not without a lot of effort on his part. Again, perhaps coming later in the 1940s, 30s, 40s, and even early 50s, the idea that the genetic material or the transmission of genetic material was mediated by proteins, not DNA. And many people who did experiments establishing that DNA perhaps was the material were ridiculed by scientific opinion leaders at the time who were wedded to the idea that proteins were the genetic material. Eventually, of course, the truth emerges, and those sort of theories move into the background, but they still can hold back advances if they come from significant scientific figures. Another very good example is, of course, one of the chemistry giants, Linus Pauling, who in the antibody field believed that all antibodies had the same amino acid sequence, and that to distinguish one antibody from another in terms of if the antigen that it would bind to was merely a way in which the antibody coiled around the antigen, forming a binding site, and that the same antibody sequence could do the same thing for a different antigen, and so on ad infinitum. And this was a instructive theory, it was called. This was something that was generally accepted by really even into the sort of late 1950s. So, advances, they go in sort of steps. There's a sort of dogma that persists, and then there's a breakthrough, and so on. And this sort of step-like progress is really still occurring now. We, of course, don't know that it's steps. We think we're on a continuous curve, but it's only when you look backwards that you can see how this occurs. I think the different areas or application areas for antibodies is a very interesting question. I think pharma is sort of has led the way in antibody research or antibody development, and in combination with academic groups for discovering application areas. But I think the clinical areas that are perhaps not so well, there are three for me that are not really well researched and this may seem a little peculiar to say oncology is one of them, because of course that's the biggest application area that everyone's working in. But I think there's a general understanding that we understand some of the biology. We understand how to make antibodies against tumor cells. We understand some of the targets on those tumor cells that we make antibodies to and generate appropriate responses, killing or stasis responses. But we don't really understand the biology of tumors. And there was a very interesting review in Nature magazine in 2011, where a group of Lambert and his colleagues drew a parallel between tumor cells in animals or humans and bacterial populations. And if you look at bacteria, which have been studied, of course, for decades, and you stress a bacterial population, the cells in the population, not all of them, but many of the cells in the population respond with a sort of survival response. So if you remove a nutrient from the bacterial population, the cells will recruit new pathways inside the cell to avoid that nutrient, but still allowing them to survive. And I think what this review was suggesting was that this is how we should view tumor masses, not as a sort of random selection of individual cells, but actually perhaps chaotic, but still a communicating organism. So that if you hit it from the outside with a toxin or an antibody or whatever, then that can trigger a survival response in other cells, which can then avoid the treatment that you're giving, what we probably refer to as resistance. So I think that's an area where we really need to understand a little better the biology, well, perhaps a lot better the biology of tumors, but that's a tough area and really requires this interdisciplinary approach that I talked about earlier. A second area that's really, I would say, it's one of the areas that I feel quite strongly about is brain disease. And generating methods to effectively get antibodies into the brain. And there have been a lot of activity on that recently, and actually a lot of very interesting work from the company Hoffmann-La Roche in Basel in Switzerland on understanding how to exploit features of the blood-brain barrier which prevent things getting into the brain to allow antibodies to cross into the brain. Because, people who have certain cancers, breast cancer, other types of cancer, typically they succumb to those cancers as a result of movement of those tumor cells to the brain. Tumors that grow in the brain, glioma, glioblastoma, which are not untreatable but very difficult to treat tumors, are not susceptible on a routine basis to antibody therapy because of the difficulty of getting antibodies into the brain at concentrations that will attack the tumor cells. So I think that's an area where it would be good to see much more effort, perhaps from pharma, perhaps from specialist biotech companies. The last area that I think is also very interesting is the treatment of the fetus. We know that the mother passes antibody from mother to fetus through the placenta. Sometimes this can be good, of course, but if the mother has antibodies that are not very good, she'll pass those also to the fetus. But there are fetal diseases, or diseases of the newborn as well. Antibody from the mother to the fetus sort of takes off, if you like, in the third trimester, just before birth. So, and that's where you get expression of a lot of fetal disease or newborn diseases. So, if antibodies that were specifically designed to treat fetal or newborn diseases prior to parturition or birth, administered to the mother, move through the placenta to the fetus and do their therapy within the womb. I think that's a really exciting area and a very difficult area to with not a great deal of effort going on in at least in the commercial world. Advice to young researchers is, there's a lot of advice one can give, and I always say that the danger with advice is that somebody might take it. But of course, there are things I think that are good for young researchers to learn from history, from looking at the past. Always, for example, keep an open mind on new ideas. Sometimes the ideas might be iconoclastic. They may be tearing down, some of the existing dogma. But you have to persist with it if you believe it and you have good experimental data. Question the scientific dogma of the time, and don't be swayed by big name opinions. Big names, of course, big scientific names are big because they've done big things, but they're not always 100% right. So if you really have a feeling that an idea or a concept is worth pursuing, then don't be put off if you can't persuade some of the big name scientists to accept the idea. It really means taking a chance on novel ideas, and I think that's important. And you might not be able to get it published. Good example of that was, albeit a long time ago, Edward Jenner in 1780, who generated the first vaccine against smallpox, wanted to publish that in the Proceedings of the Royal Society of London. And the reply he got from them was, "This is too preliminary." So he actually wrote and published his own magazine in which it was circulated and generally eventually accepted in the scientific community. I don't think we can do that today, but it's certainly a good example to follow. I think the last thing I would say is that always be true to your data. Never hide experimental data that you believe might disprove your ideas, and really maintain, as far as you can, 100% honesty in the way you present your experimental evidence, because in the end, that's what's going to be important. In terms of the IBC conferences on antibody engineering, of course, it was exciting to be here this year, the 25th anniversary. And I remember some of the early meetings and the excitement, because they were smaller then. Of course, the excitement of new ideas and new discoveries coming out. Of course, it's a larger meeting now and a vast amount of work going on. And I think it's probably is putting it a bit too mildly. It's certainly, I think, the most important antibody meeting on the calendar. I guess there are maybe upwards of 800-900 people. I'm not sure how many, but the diversity of the subject field now means that parallel sessions and multiple topics really attract a large group of immunologists, not just doing core antibody work, but other aspects of biology, target discovery, and so on. So I think it's a great meeting, and although I haven't been for some years, it was a great honor and pleasure to come to this meeting and talk about the history of the field. And I hope to attend next year, actually, in a slightly different capacity, because hopefully we'll be forming a new company with some new ideas to present.